Next topic is post-fertilization structures and events in plants. Post-fertilization means after fertilization, after double fertilization. We already discussed what is double fertilization, and it is a common phenomenon or a specific phenomenon found in angiosperms or flowering plants that we already discussed. Double fertilization. After double fertilization, what are the post-fertilization changes in a flower? Here, post-fertilization structures and events. In this topic, we can discuss development of endosperm, embryo, seed, fruit. Here, we can discuss first two, development of endosperm and development of embryo, then different types of embryo. Then here all these are development of endosperm, embryo, seed, fruit. All these structures and developments together called post-fertilization events in a plant. We can discuss first one, development of an endosperm. What is endosperm? That we already discussed before in the another topic. Endosperm, that is in double fertilization. Endosperm is the product of triple fusion. Which fertilization? Triple fusion. Endosperm is the product of triple fusion. Triple fusion means fusion of three nuclei, one male nucleus and two polar nuclei found in the embryo sac. Triple fusion, product of triple fusion is endosperm. Then so endosperm is a triploid structure. It is triploid. Then endosperm is nutrient rich in nutrients. Endosperm is rich in nutrients. Rich in nutrients. Then function of the endosperm provide. It is rich in nutrients, so endosperm provide nourishment to dash. It provide nourishment to embryo, developing embryo. So that is what is the function of endosperm. Endosperm is rich in nutrients. It provide a nourishment for the developing embryo. These are the important points about the endosperm. What is endosperm and the structure of endosperm and also function of the endosperm. Then we can discuss development of the endosperm. Development of endosperm. Then endosperm development after triple fusion. The first product is a nucleus called a pen. What is pen? Primary endosperm nucleus. That nucleus is known as primary endosperm nucleus. Then this is a triploid structure, triploid nucleus. Triploid nucleus. This primary endosperm nucleus undergo free nuclear division. This is a nucleus, free nuclear division. Primary endosperm nucleus undergo free nuclear division. Then form an endosperm called a free nuclear endosperm. That endosperm is known as free nuclear endosperm. Primary endosperm nucleus is the first product of triple fusion. Primary endosperm nucleus undergo free nuclear division and form thousands of nuclei. Then that endosperm is known as free nuclear endosperm. Here is an example of free nuclear endosperm. Tender coconut water. Example of free nuclear endosperm. Very familiar. Tender coconut water. Tender coconut water means karikin vellam. Contain thousands of nuclei. Free nuclear endosperm. That is what is tender coconut water is an example of free nuclear endosperm that contain thousands. That is rich in nutrients. Okay. Then that free endosperm undergo cell wall formation. Next is in free nuclear endosperm, cell wall formation, then form an endosperm called cellular endosperm. Next, free nuclear endosperm undergo cell wall formation or cellularization, then form cellular endosperm. That endosperm is known as cellular endosperm. Then next, final product is mature endosperm. Last, it become mature. That is what is the development of endosperm. You can write like this in a flow chart. Development of the endosperm start from primary endosperm nucleus. It undergo pre-nuclear division. Pre-nuclear endosperm. Then it undergo cell wall formation, forms endosperm. Cellular endosperm undergo changes and become mature endosperm. That is the development of endosperm. Okay. Then based on endosperm, there are two types of seeds. Based on endosperm, there are two types of seeds. Two types of seeds based on endosperm. First one is endospermic seed. Endospermic seed. As we all know, what is endospermic seed? Seed with an endosperm. Seed with an endosperm is known as endospermic seed. It is also known as albuminous seed. It is also known as albuminous seed. Endospermic seed is also known as albuminous seed. Albuminous seed means same thing, seed with an endosperm. Example, all monocot except all monocot except orchids. Orchid is an exception. All monocots are having endosperm, but orchid is not having endosperm, but orchid is a monocot. Example clear? All monocots. Monocot means rice, wheat, maize, coconut, bamboo. All these are examples come under monocot. All monocot except orchid. Orchid is also an example of monocot, belongs to monocot class. Albuminous seed, example, all monocot. Then second type of the seed, non-endospermic seed. Non-endospermic seed. Non-endospermic seed is also known as non-albuminous seed. Non-albuminous seed. Non-albuminous seed. Non-endospermic seed means seed without an endosperm. There is no endosperm. That is also known as non-albuminous seed. Example, all dicot except castor. Castor is an exception. Castor means avanak. Castor is an exception, but it is dicot. All dicots are not having endosperm. They produce non-endospermic seeds, but exception is there. Castor seeds are having endosperm. Clear the difference? Then listen, non-endospermic seeds are called so. Why? There is no endosperm. That endosperm is completely utilized for the development of the embryo. That's why endosperm is not there in the mature seeds. That is what is the reason behind it. The seeds of the dicot are non-endospermic because complete utilization of the endosperm for the development of embryo. That's why they are non-endospermic seeds. Endosperm is not there in the mature seeds. But in monocot seeds are known as endospermic seed. They are having endosperm. That means endosperm is not completely utilized. It is partial utilization of the endosperm for the development of embryo. That's why endosperm is retained in the monocot seeds. That is the difference between endospermic and non-endospermic seed and the examples. All of you clear? Then next development in post-fertilization events, development of embryo. Next is embryo. Development of embryo. What is embryo? Embryo is the product of syngamy. Embryo is the product of syngamy. Embryo develop from zygote. It is developed from the zygote. Then embryo become new plantlet. It produce new plants. New plantlets or seedlings. New plantlets are arise from the embryos. Embryo is the product of syngamy. Then it developed from the zygote. Then it become embryo develops into or produce new plantlets. That is what is embryo. Embryo produce. Where are they situated or where are they produced? This embryo is present at micropylar end in the egg apparatus within the embryo sac, where before that zygote is situated. Then zygote convert into embryo. That is what is embryo. Then we can discuss early stage development of the embryo. That is what is called embryogeny. What is embryogenesis? Embryogeny. Embryogeny means early stage development of the embryo. Early stage development of the embryo is known as embryogeny. In the first chapter you have learned embryogenesis. What is embryogenesis? Embryogenesis means development of embryo from the zygote, but it is different. Embryogeny. Embryogeny means development, early stage development of the embryo. This is common. This is same in both monocot and dicot. All the plants are having same embryogeny. Starting from, you can write embryogeny in the form of a flow chart. Embryogeny common for both dicot and monocot. Early stage development start from zygote. Zygote is diploid. Zygote become pro-embryo. Zygote become pro-embryo. Then pro-embryo become globular embryo. Pro-embryo become globular embryo. Globular embryo become heart-shaped embryo. Globular embryo become heart-shaped. Then heart-shaped embryo become mature. Heart-shaped embryo become mature embryo. This is this arrangement. This is called embryogeny. Embryogeny means early stage development of it start from. It is common for all plants, both monocot, zygote. It start from zygote. Zygote. This stage of the embryo is called pro-embryo. Then pro-embryo become globular embryo. Globular embryo is there with a suspensor. As neck-like structure is there. Diagram is in your textbook. I will tell you which page number and also the figure number. You can draw that also. Globular embryo convert into heart-shaped embryo. Then the final product is mature embryo. That is what is called embryogeny, early stage development. Then we can discuss two types of embryo. Two types of embryo, dicot and monocot embryo. Dicot and monocot embryo. We can discuss. It is very here dicot and monocot embryo. What is the difference between? I already wrote the points are here. The difference between difference between dicot and monocot embryo. As usual in the class, you are following the same thing. Difference in the same note down. All of you note down the difference in the table itself like this. One column for dicot embryo and another column for monocot embryo. Then points. First point, cotyledon present. Cotyledon means, what is cotyledon? Cotyledon seed leaves are called cotyledon. Then cotyledon stored reserve food materials for the seed. Then there are two cotyledon present in dicot. In monocot only one cotyledon. That cotyledon is known as scutellum. What is it? Scutellum. What is scutellum? Scutellum is a single cotyledon. It is present in monocot embryo. Both are having cotyledon. One is having, dicot is having two cotyledon. Monocot is having only one cotyledon. That is what is called a scutellum. Scutellum is situated at a side, lateral side. Here the diagram of monocot embryo. This is what is scutellum. It is situated at the lateral side, a side of the embryonal axis. Then here 2 cotyledon present in dicot embryo. Then next embryonal axis present in both. This arrow mark shows same point in this column. Embryonal axis present in dicot and monocot. Embryonal axis with plumule and radicle, upper part and lower. Plumule becomes shoot and radicle become root. Then next epicotyle present. Epicotyle, the term epicotyle. Epicotyle present in both dicot and monocot. What is epicotyle? You can note it down in the same point itself. Epicotyle means the part or the region above the cotyledon. Region above the cotyledon is known as epicotyle. Here somewhat here epicotyle, hypocotyle. Okay, epicotyle present above the cotyledon, that become, I wrote here, shoot. Epicotyle, that means it convert into shoot. It becomes shoot. Then hypocotyle present in both dicot and monocot. Hypocotyle present. Hypocotyle means the region below the cotyledon. That is the difference. One is above the cotyledon, other one is below the cotyledon. Hypocotyle present below the cotyledon. Below the cotyledon means near to the radical portion. Radical. Okay. Then coleoptile, epicotyle, hypocotyle present in both dicot and monocot. The next structure, coleoptile. Coleoptile. P represent plumule. That you have learnt already. Coleoptile absent. Coleorhiza also absent in dicot embryo. But these two structures present in monocot embryo. What is coleoptile? Coleoptile is an undifferentiated sheath-like covering that to protect the plumule part or the shoot part. Okay. The coleoptile means an undifferentiated sheath-like structure cover the plumule. That is what is called a coleoptile. Is present only in monocot embryo, absent in dicot. Coleorhiza means R is here. Coleorhiza. Coleorhiza absent in dicot. Coleorhiza present in monocot. What is coleorhiza? Undifferentiated sheath-like structure that is cover present in the radical portion of the monocot embryo. That is what is coleorhiza. These two structures only found in monocot, absent in dicot. Then next two diagram. These diagrams in the page number 35, figure number 2.14, A and B diagram. After this type, different types of embryo and the difference in the same column, you can draw the diagram also. Very important diagram. This diagram, grass embryo. In the textbook, it is not noted like that. Grass embryo. Grass embryo means grass belongs to monocot class. So, monocot or grass embryo, the same embryo you can draw. Okay, scutum at a side, then coleoptile here label, then there is an extra outgrowth is there, epiblast, then radicle, coleorhiza, here root cap. All these are the labeled parts, different parts of the monocot embryo. Dicot embryo is very simple than monocot embryo. 2 cotyledon, then hypocotyle region, plumule, here epicotyle region also, then radical, root cap. Very simple diagram. Dicot embryo. Monocot embryo is with many different parts are there. Okay, that is what is the difference between dicot and monocot embryo. Clear? This topic, post-fertilization changes, endosperm development and embryo development. In the next video we can discuss seed and fruit also. Then here one more diagram note down in the textbook. Embryogeny. Embryogeny page number 34. These are the diagram. Page number 34, figure 2.13, B diagram. B diagram means here zygote, starting from zygote. Second diagram is not labeled. You can label it as pro-embryo, that flowchart. In the flowchart itself, after zygote, pro-embryo is there. This is globular embryo. Then next structure is heart-shaped embryo, then mature embryo. This diagram also you can draw it in your notebook after embryogeny. Then write different types of dicot and monocot embryo, its difference, then the diagram. Okay.